class, the relationship between enzymes and the factors that affect their rate of reaction were taught. This dynamic was then exhibited through the pineapple jelly lab where the enzyme bromelain was observed. To make jelly, there must be gelatin, which is made from a protein collagen that has been formed through the addition of hot water and then letting it cool. In fresh pineapple, bromelain, a protease enzyme for the hydrolysis of collagen, is present. This enzyme can survive temperatures up to 70 degrees Celsius. But canned pineapple, in order to kill the bacteria that would shorten its shelf life, has been heated to temperatures upward of 100 degrees Celsius. From this information, the lab was derived on the basis of what type of pineapple would create a more solid jelly, with the independent variable being type of pineapple, canned or fresh, and the dependent variable being the solidity of the jelly. Additionally, some control factors include the time left to set, the temperature of the water, the amount of jelly solution, amount of pineapple, environment, and type of jelly powder. The hypothesis is as follows. If both canned and fresh pineapple's abilities to create pineapple jelly were tested, then the canned pineapple will make a more solid one due to its lack of the enzyme bromelain. This is because jelly is derived from protein collagen, so the presence of the enzyme bromelain breaks down the peptide bonds of protein through hydrolysis and will interfere with the making of jelly. Canned pineapple would not have bromelain since it's been heated past its optimal temperature and therefore the enzymes have been denatured. As such, the canned type will yield a more solid pineapple jelly. Following the outlined procedure, gather the lab equipment. 2 400ml beakers, a 100ml graduate cylinder, a stirring rod, a petri dish, plastic wrap, paper towels, an electronic balance, and a wax pencil. As for the ingredients, this consisted of diced fresh pineapple, canned pineapple, jelly powder, hot water, and cold water. Next, to prevent confusion, label the beakers using the wax pencil with the labels canned and fresh. Then measure 42 grams of the jelly powder in a petri dish and place into any one of the beakers. After, using a graduate cylinder, measure out 125 milliliters of hot water from the kettle. Pour the water into the beaker containing the powder and mix using a stirring rod until all the powder dissolves. Next, with the graduate cylinder, measure out 125 milliliters of cold water from the tap. Pour the cold water into the same jelly mixture and stir well. After that, again with the graduate cylinder, measure out 125 milliliters of the jelly solution and place it into the other labeled beaker. Then, using the electronic balance, measure 60 grams of canned pineapple in the petri dish and place the pineapple into its appropriate beaker. Using the stirring rod, gently stir the jelly in the pineapple mixture. Following this, wash the petri dish and stirring rod thoroughly as to prevent cross-contamination. After, using the electronic balance again, measure 60 grams of the fresh pineapple in the petri dish and place the pineapple in the appropriate beaker. Using the stirring rod, gently stir the jelly and pineapple mixture. Finally, cover both beakers with plastic wrap and place them into the refrigerator. Clean up your lab station and equipment and put them back into their appropriate places. After approximately 24 hours, the state of the jellies were observed. The canned pineapple jelly was fully developed into a firm solid, whereas the fresh pineapple jelly was in the same state as it was when it was first left to set, liquid. Both types of pineapple had soaked up the jelly solution and were tinted the same red color. Both beakers smelled the same as well, sweet and tangy from the combination of pineapple and sugar from the jelly powder. The only observable difference was how the fresh beaker seemed to have a much larger volume than the canned. The most vital point of contrast, though, was ultimately how one was successful in creating a firm, solid pineapple jelly, while the other didn't work to any degree. To thoroughly expand on one experimental error, many factors were not consistent despite being controlled variables, for example, the temperature of the water. Though the water was taken from the same faucet for cold water and the same kettle for hot water, there's no thermometer used to measure the exact temperature, nor was any temperature specified for the water. And this matters greatly because the whole point of this experiment was to test the difference between canned and fresh pineapple. But if the fresh pineapple was submerged in hot water around 100 degrees Celsius, temperature that was stated which the canned pineapple was treated at, for long enough, then that would denature bromelain. This is very similar to an actual method of canning called the water bath method. This would have yielded two perfect or very similar jellies, as both pineapples would have been basically the same. It is also not hard to get water to 100 degrees Celsius, considering that is the boiling temperature of water. Thankfully to canned pineapple, fresh pineapple should be submerged in hot boiling water consistently for 15 to 20 minutes depending on the amount of fruit. This was obviously not done, as within less than 15 minutes the cold water was added to the solution. But that is only specific to this experiment. So moving forward, the temperature and time frame of when the cold water would be incorporated should be specified within the procedure. To conclude, the hypothesis was confirmed to be correct by the experiment, and canned pineapple is indeed better at making a solid jelly than fresh pineapple.